Hello students. So today we are going to learn about vectors. So this is a part of rDNA technology. So let's learn about vectors. But before that, first understand what is vector. So vector is a type of carrier molecule which carries our desired gene of interest in the host cell. So now there are two type of vectors. One is cloning vector and one is expression vector. So what's the difference between these two vectors? Let's understand it. Cloning vector is used for multiplication of gene, which means that if we want to multiply the gene, if we want to replicate the gene, then we will use this cloning vector. Whereas expression vector will be used when we need to express our gene, express our gene into protein. At that time we will use this expression vector. Now you understand it? Okay, let's move on. So now, for a good vector, what should be the properties of the vector? So here are some properties of the vector. Let's understand each one of them. So we will understand these properties one by one. Let's understand it. So first property is selectable marker. What is selectable marker? Uh, for example, this is the plasmid PBR322. PBR322. So here the selection markers are tetracycline and ampicillin. They are used for screening. If we want to check whether our host cell is transformed or not, at that time the selection markers are used. Now, second property is self-replication capacity. So a plasmid should have a self-replication capacity so that it can self-replicate in the host cell, such like OD site. From this OD site, the plasmid, or we can say vector, will replicate itself. So the third property is compatible with multiple host cell. A vector should be compatible with multiple host cell. For example, if we are using one vector, then it should be compatible with number of host cell. Now, fourth property, multiple cloning sites. A vector should have multiple cloning sites. For example, if we see in PBR322, then there are multiple cloning sites. In the tetracycline resistant region, there is HIND3 site, PAM-H1 site, SAL1. In the ampicillin resistant region, we have SCA1, PVU1, and PSC1. So these are the cloning sites, or we can say multiple cloning sites present in PBR322 vector. Now, the fifth property of good vector is that the size of the vector should be small. Why it should be small? Because if the size of the vector will be big, then it will not enter in the host cell. So it should be small enough that it enters into the host cell and the host cell will get transformed. Now the sixth property is should be stable for longer generation. The vector we are using, it should be stable for multiple generations. It will not get degrade. It should be stable enough for a long time. Now the seventh property is high copy number. What is high copy number? That if we are using vector, then that vector should have high copy number. Means it should replicate number of times, or we can say um, if the vector has high copy number, then it will copy itself too many times. So it's a good high copy number. Now, high insert size for gene. What is high insert size for gene? So if we look in this, PBR322, then these are the insert insert regions where we can insert our desired gene of interest. So if the size of the insert is small, then it may be possible that a gene will not be fitted in the vector molecule. So the size will be um, maximum enough so that it can insert our gene of interest. Now the last property is construction of genetic library. So uh, it should promote the construction of genetic library. If we want to study some organism genome, then we should be able to construct a genetic library in vectors. So I hope you understand the properties of a good vector. That what are the properties of a good vector? Let's fastly review it. That it should have selectable marker for further screening. It should be self-replication, means it should have self-replication capacity, which is OD site. It should be compatible with multiple host cell. It should have multiple cloning sites. The size of the vector should be small, should be stable for longer generation, high copy number, high insert size for gene, and construction of genetic library. Okay, so now let's move on. So now let's understand the types of vectors. How many types of vectors are there? So generally four types of vectors are there: bacterial vector, yeast vector, plant vector, and animal vector. We will understand all these vector individually in some video afterwards. Now, nomenclature of plasmid cloning vector. So let's take an example of PBR322. PBR322 conforms with standard rules for vector nomenclature. So now what are those rules for vector nomenclature? Let's understand it one by one. So here P stands for plasmid. BR stands for in PBR22, stands for Bolivar and Rodriguez, which is two researchers who developed PBR322. Generally, BR is identify laboratory in which vector was originally constructed. Now, what is this 322? 322 distinguish plasmid from others developed in same laboratory. Means that in the same laboratory, many types of plasmids are developed. From them, one is this PBR322. There are also plasmid called PBR325, PBR327, and PBR328, etc. Generally, 322 stands for that after this many means they had done this many trials. After 321 trial, the 322th trial is a successful vector, so that's why they have named it PBR 322, where 322 stands for the successful vector after having 321 trials. So this is for today, guys. We will learn now the vector individually in the next video. Till then, please like the video, share it, and subscribe it. Thank you.